streaming. Meanwhile,
okay, great. Just starting now, I guess.
Mohammed, can you share? Okay. Yeah, okay. Looks good.
oh, the camera. Good one. What is it? Oh, I need to adjust the camera. About now? Okay. About now? Good.
Fine.
Already shared. Can you confirm from Zoom? Perfect.
Yeah, you can start.
Don't touch. Okay. Finally, we got it ready. Any problem? You can hear me, everything fine, right? Okay, perfect. How? Why? Why? Okay, so after all the hassle, managing the live streaming, the Zoom meeting, finally we are starting. Good afternoon, everyone. I'm Mohammed Alser, and today I will be talking about totally different flavor, which is intelligent genomic analyses. So I'm lecturer and senior researcher at Safari since almost 4 years. I got my PhD from Bilkent University, Turkey, where I was working on bioinformatics topics. I got the best PhD thesis award from IEEE Turkey. And this is my Twitter, where I share things about bioinformatics and personal views. I'm working mainly in bioinformatics, computational genomics, and metagenomics. Okay, so for today, I will cover different topics, starting from motivating you why we should work on this area, why as computer scientists or computer architects, we need to discover things related to biology and our health. And then I will define intelligent genome analysis, why we need it to be intelligent, why not just accelerating, and that's it. And then I will define the barriers that we need to pave the way for them, or for accelerating or making intelligent genomic analyses. And then I will show you the work that we did at Safari Research Group at ETH to accelerate different part of the pipeline. And toward the end, I will tell you where we are going next. Okay, what is genome analysis? So before I start, this is our recent paper surveying the entire topic of intelligent genomic analyses. I would recommend everyone to read this paper carefully and make it close to your heart and see what are the potential projects that you can work on during your master, PhD, or thesis project. or even semester project. All right. So when you would like to analyze something, basically, what you would like to get, what you expect to get. Is it just mere data, just some information scattered, or you would like to get insights and observations? And this is Richard Hamming definition. Richard Hamming, who invented the error correction for network, and he is well known for Hamming distance, which is a measure between two strings or two different things, and how you measure the errors or the differences between them. And likewise, we need really to gain these insights and observation much more efficiently than ever before. Why is that? For two main reasons. The first one, genomics is one of the major generator for huge data. It's no longer big data. It's really much bigger than big data. The second one, we are already witnessing the start of the angstrom era, where the transistor size now is less than 1 nanometer. And for that, we really need to start thinking about making genomic analyses more intelligent and how we define genomic analyses. So this is basically the definition of data analysis or data mining, which is the science and art of revealing previously unknown and potentially valuable information or knowledge from data. However, to make it intelligent, we need to respect some constraints while meeting functional, performance, energy consumption, cost, and other specific goals. So we cannot worry about only the execution time of the analysis. We need to worry about many other specific things that I'm going to mention next. And before we start, we need to set the stage for genomics, since you don't have background, or I don't expect personally that you have a background in this topic. If you look at this picture, what you can observe. People with different traits or different phenotypes, right? Things that you can clearly and directly observe just by looking to the face. They are different in hair color, face color, or skin color, eye color, and so on. Even the body mass, right? What makes them different is the DNA. So the DNA is the organism complete set of genetic information or genetic instructions. If you enable one of these instructions, then you're probably going to enable something to observe from inside and outside. As a computer scientist or computer architect, what we need to worry about is this. We don't deal with the molecules or the DNA itself, but we deal with the text format. And this is representing our DNA or our genome. How is that? So the DNA is represented by 4 DNA alphabet, A, C, G, T, only those 4. What are these? These are the 4 chemical molecules, adenine, guanine, cytosine, and thymine. And that will be the full DNA just containing these 4 letters. Now, how about this? What is this tower? Prime Tower in Zurich. Excellent. All right. Now, if we print the human DNA on A4 paper, and then we use very small margin, very small spacing, we use font size of 12, how many papers we could fill with the human DNA, single human genome? If we print all the alphabets in your DNA, A, C, G, T, just print it out. You get exactly the same length as this tower. It's a huge workload. That is for single human. If I want to analyze all people, the genomes of all people in this room, then you can think about the huge workload here. So the size is about 3.2 billion characters. Now, if we talk about other species, this is a virus, is a few thousand characters. Bacteria normally much longer than that, so a few million characters. And by the way, this virus can infect this bacteria. Human is midway, 3.2 billion characters, of course, varies from 1 to 1. With the onions has longer DNA than ours, 5x longer. this Japanese flower about 50x longer than our DNA. So it really varies from species to species. All right, so how we start analyzing our genome? First, you need to purchase some of these kits or simply go to the hospital or to the lab. They will take a blood sample and then start analyzing it. What they will see, 23 pairs of chromosomes. One strand in any chromosome coming from the mother, the other one coming from the father. So you're inheriting genetic instructions from your parent. But there are also some other information that are not inherited from the father or the mother. We call them de novo variations, something that very unique only to you. And this is what we are trying to read, basically, the content of your chromosomes. This is under electron microscope. So you can see the chromosome, how it looks like in real life. This is inside your cell. So inside any cell in your body, you will have the exact same shape of those. Those are 23 pairs of chromosome. Do you know this lady? This is the actual lady. This is the actress or the movie that telling the story of this lady. How many of you watched this movie? The Immortal Life or Henrietta Lacks. So this lady was going to a hospital for some regular checkup, and then they figure out that she has a cancer, and then the doctor kept some of her cells and then distributed her cells to many hospitals to study the genetic cause for a cancer. Without her consent, she was not agreeing on that. And then around 50 years later, all the research groups start studying her genetic material. They discover even the genetic material of her sibling and her family members. And that was a red flag showing the importance of not giving your DNA or your information to someone else who can reveal more than what you can think about. So you can watch this movie and learn more about this. This is very famous cells called Henrietta Lacks. All right, so once you get this genetic information or the DNA or the sequence of genome, the DNA basically, which is the genotype. So you can think about it as a cookbook where you have a lot of recipes, and any of these recipes going to cause you making the food, right? And that's exactly what we have. So DNA is going to transcript it into RNA, and then RNA translates it into protein. And that protein is what you observe, what cause all the phenotypes that you can see. Now, as I said, in any cell in your body, it has the exact same amount of information, regardless whether the cell is heart cell or heart tissue or leg cell or whatever tissue you have in your body, they are having exact same genetic information. The only difference is some of them activated, some of them is not activated, basically. And when you activate something or something changed inside your genome, it might cause a disease. Some of these genetic differences, we call them SNP, single nucleotide polymorphism. It means single change in your genome, just single characters changing and causing that disease. And those studies, normally we try to figure out what is the cause for that disease, or what is different between me and you, where, for example, I hold some disease, you don't hold that disease. Again, it doesn't have to be a disease, it could be just a phenotype difference, like eye color, right? It goes also by genetic differences. Now, all of you are smart people. We have this part of your genome. These are multiple individuals, and we are studying blood pressure disease. So we got some readings for the blood pressure, and we observed that for those people, it's always high, but for those people, it's low. Now, think about which character, which location was causing the blood pressure disease, just by looking to the strand of your genomes. So if you look at SNP2, you will see it's really not correlated to the high blood pressure, right? Could be G, it could be A, and so on. But if you look at the SNP1, whenever it's C, directly can be correlated to high blood pressure. And that's what we do as GWAS studies, Genome-Wide Association studies. So we got a group of people who are healthy, they don't hold the disease that we are targeting, and another group of people who hold that disease. And then we compare their genomic information together. We draw what we call a Manhattan plot, where it show the frequency of those SNPs. For example, this individual at chromosome 2, they have this variation. And this variation is happening more often than the rest of the SNPs. So we draw a threshold and we said, all these SNPs are differences between this group and that group. The more people in both groups, and the more fine grain we select these people in the group, then we successfully can detect what SNP is associated with which disease. There are many other studies, not using just the DNA, but using other genetic information. So this is one example of it. If you're interested, feel free to read it. Now coming to real life practice in hospitals, this is one of the perfect. This is one of the links. You can open it in your personal computer. It will direct you to this database called OpenSNP, single nucleotide polymorphism. What it tells you, you'll get exactly the same window as here. So you got the chromosome 19, the position at this chromosome is this, and then you'll get this information. It's telling you if you got the T from your father, T from mother, then there is 20 to 25% that you may get better or to treatment to hepatitis C. But if you got C from the mother, C from the father, then 80% that you may respond to a treatment. So based on that, the doctor can prescribe different medication based on your genome and can tell you may get better with this treatment, but not the other treatment. Although maybe other people have the opposite thing. And this is very useful for personalized medicine. All right, so we were talking about single nucleotide polymorphism. Now let's talk about really large variation happening in the genome. It's not just single character changing or switching from A to C. By the way, I can hear you myself. I don't know if it's real loud. Is the voice good? Okay, perfect. Yeah, so if you consider autism, for example, you will observe a deletion of this much, 500,000 characters deleted from the genome for people who have autism. Obesity, the same thing. In underweight, which is totally opposite disease, you will see the opposite effect, duplication of 500,000 characters. So 2 regions are duplicated in this chromosome. And this is the paper studying this disease. So in the second line, if you're interested, you can check these links. And that's what we call structural variation. It's no longer single nucleotide polymorphism or small variation. We call them structural variation because they are really large. Any variation longer than 50 characters, we call them structural variation. And this is a very good paper to read about this topic. All right, so hopefully now we set the stage, all the information needed to define the genomics. Now, what is intelligent genome analysis? So we want first fast genome analysis. So we are talking about the bandwidth, and then we want population scale genome analysis, because as we mentioned in GWAS studies, we need 2 groups of people, one healthy and one unhealthy. So one scalability, it's not about how long you can achieve one analysis, but how many analyses you could do in the same time. If one analysis is taking 10 minutes, then how about 1,000 analyses? Is it 10 times 1,000, or is it really still 10 minutes? We want to use intelligent architecture because we don't want to build huge clusters for hospitals to start doing the analysis on a daily basis. Apart from the energy efficiency, we don't want to have huge cooling systems for these clusters in order just to do genomic analyses in the hospital. DNA is a valuable asset, so we don't want to release it or make it public for everyone to read, because it's not about only you, but it's about your family members, your parent, and your relatives. They can reveal much more about them. And it might cause any issues because health insurance companies might block you from the policy once they know that you have this disease or that disease. We don't want to give errorness analyses, and that is about the accuracy. I don't want to tell you that you have this disease, but you don't have it. Why we should care? Why intelligent genomic analyses is important? Why not just talking about the speed? How many of you watch this movie, Gattaca? Very old one. 1, 2, 3, 4, 5, 6, 7, 10, 11, 12. Okay, perfect. Great. How many of you watch this recent movie, Tomorrowland? 1, 2, 3. You don't watch movies, guys? 7. Okay, perfect. So in Gattaca, you'll see something like that. So they have the entrance to a building. You just swap or touch your finger and then identify you based on your DNA. So it will read your DNA, and then what could be better than your DNA to identify you? Nobody can hack it, right? It's really different than fingerprints, for example, or biomedical fingerprints. And to do that, you don't want to wait like 1 day just to get access to the building, right? You want to do it in few seconds, really few seconds. And that is our target, to do fast genome analysis in mere seconds using limited computational resources. We don't want to build a huge compute centers just to do the analysis, because you may need to do that in remote locations where you don't have access to internet. For example, during the Ebola time in Africa, there was a huge need to do the analyses in very remote areas where you don't have access to cluster, you don't have access to internet. Then good luck doing the analysis. Talking about hospital, there is a huge need to do the analysis quickly and scalable. So for critically ill infants, you have two options when you go to typical hospitals. Either to do the whole genome analysis in 2 days or 5 days. If you want to do it in 2 days, you will pay a lot. If you want to do it in 5 days, you can save something, but maybe the baby won't survive. And if you do whole genome analysis and based on some personalized medicine databases, you could discover what was causing the disease or why the baby is really sick. Then you could save morbidity, reduce hospital stay length, and inpatient cost. Those are very important factors. What is so special about infants? Normally, doctors are confused about the symptoms, so they don't know. All babies will have fever. Then what causes the fever? They might not know what is the symptoms or the disease that causing this fever. So that's why they have to read the genome. And most of the cases for critically ill infants is genetically disorder. So they have to discover that and then prescribe the medication based on your DNA. And that's why in UK, for example, in 2019, they start to do whole genome sequencing for any admitted critically ill children. So you can think about the scale. That is UK-wide. Now, if you try to do population scale genomics, for example, you want to study all the people living in Zurich. You want to see what kind of disease they have so that the government care about the people, for example, and they want to make all this medication available for them. So one of the study here, it needs about 4 million CPU hours to process about only 50,000 individuals, or just to read their genome and try to identify the variation. Think about if one scale this to countries or continents or the entire Earth. This is during the disease outbreak. This is Ebola in Africa, where you need to do that massive scale testing. This is for COVID-19, where you take DNA sample rather than taking PCR, because PCR will work only when you know what you are targeting, what you are looking for, because you need the reference, and then you check for PCR. If there's a chemical reaction with the reference, then it means you have COVID. But if there is no chemical reaction, then you don't have COVID. That is PCR test, in short. However, when you don't know what you are targeting, for example, the first year of COVID pandemic, We don't know what we are looking for because we don't know the virus genetic information. In this case, we have to use genome analysis. We have to sequence the virus, knowing the series of the DNA strand, and then we can do the PCR test. There are a lot of studies done by the government. They regularly check the subway station, for example, or the crowded places in the world. What they do, they try to identify what organisms presented in a given environment and how abundant are they. This is another recent study. You can check here. They are trying to identify all viral information in the entire world, or all viruses. And what they need to do that, about 28,000 virtual Amazon Cloud CPUs, just to do the assembly for 3,500 samples or assemblies. That's a huge workload. That's a lot of energy taken out just to do this analysis. This is another study done in 2015. Here's the link, where they were targeting only New York subway stations. And they took about 1,500 samples. Hopefully the people on the site can see the slide. And they tried to identify and see what is going on in New York subway. And this is what they find. 50% of the things they study, they don't know what is it. They never studied before. About other 50% is bacteria. Another study tried to check the microbiome in 60 cities. Yes, please. Microbiome is the microorganisms, viruses, bacteria, and so on. So we try to identify what viruses exist in a place. They take sample from everything in any subway station, including ticketing machine, door handles, the seats in the train, everything related to the train station. Same thing here. There are a lot of pictures. If you go to this paper, there's a website for the tool. You'll see the picture taken even around ETH, because one of the cities, including this 60 cities study, was Zurich. And you'll see that they were taking sample from the screen of the ticketing machine. Because if there are viral infection over there and you touch it with your finger, then good luck. If you touch your eye or something, then you'll get the infected directly. So that's why they were trying to build a map to see what kind of viral information or bacteria around the world. Is it different? Is it unique to each city, for example? That's a huge scale, by the way. You can see 5,000 sample, and you need to process 5,000 sample using computational resources. Going back to the study done in 2015 in New York subway station, what they find Yersinia pistis. This is one of the bacteria that caused a huge, massive death in Europe in about 40th century. And to see something deadly in a subway station, very crowded place as in New York, it's really red flag for everyone. It was in the breaking news for a few weeks in the US. And once they go back to traditional method, they couldn't find Yersinia pistis over there. That was a false alarm. And then people start suspecting or doubting the benefits of bioinformatics. Some people even call it a failure of bioinformatics, because they were using computational tools that were not accurate, and then they were showing that you have this bacteria in your sample, but it does not. That's why speed is not the only important metric here. You could build very efficient Fpga accelerator for these tools, but you could ruin the accuracy, and that is not good. That's why we need intelligent genomic analyses and not only fast genomic analyses. This is one of the studies that we participated in, published in Nature Method, about evaluating the existing tools and see what kind of new species that we can discover. This is another tool that we developed, Metaline, MECOP. And now, talking about reliability. Think about you are in this situation and some failure happening in the cockpit of the flight. What will happen? Yeah, then good luck, right? You need to restart the computer, maybe, right? And then see what happened. Same thing for genome analysis in outer space. This is in a space shuttle. Why do you need to do genome analysis over there? Because some of these shuttles stayed for about 6 months or a year, right? They cannot send the sample they collect from other planet to the Earth because the sample might get damaged. The shuttle itself might get damaged, right? And then you lose the sample, you lose everything you were targeting. But also these samples could be contaminated with other infection or other viral information, maybe from the inside the shuttle itself, and it's not related to the planet. So it will be always beneficial to do the analysis right away. Once you collect the sample, you do the analysis and then send the genomic data or whatever you discover to the Earth or to the people to do further discovery. And this is, by the way, very challenging over there because there is no gravity. And then there will be air bubble inside the sequencing machine, the machine that's going to read the genomic information. And once you find air bubble in the machine, then you'll get a lot of error in the output. You get low quality data. And then that's going to introduce more additional computational steps just to correct these errors. And those issues you may not, or they may not be very important to you on Earth, but they will be important in outer space. So we are still talking about intelligent architectures for genomic analyses, how we can enable data movementless pipeline. We really don't need to move the data from the machine that reads your genome to the main memory or to the storage, and then to main memory, different level of caches, then to the CPU or FPGA and GPU. That's a huge data movement. As I said, your single DNA strand or information about 3.2 billion characters. That is something huge to be moved between different components. This is one of the real machine. This is just a toy example. I'm not sure how much the accuracy these days for this device. This is a real device from Nanopore, Oxford Nanopore Technologies. And it attached to the iPhone where you do the sequencing here. So you read the genomic sample over here, then send the information to the iPhone, and then you can do the analysis over there. So far, nobody's using this for real life because the accuracy is not that good. And the speed of the analysis at iPhone is not that good as well. So, but this is the ultimate goal. We want something really portable where we can do the analyses in very small device without accessing huge clusters. There's another work talking about the privacy. It's very important to keep your DNA or genomic information apart from other strangers, let's say, because in this work, we figure out that there are a lot of ways where you can infer a lot of information from your DNA plus social media. You can figure out from your picture what eye color you have, what hair color, and then from your siblings, and then you start building your DNA as if you have the full DNA content. There are a lot of nice stories here, how you can reveal and what you can reveal from this paper. That's why some of the companies start offering privacy-first DNA testing. So privacy here is very important. It's still questionable since this company is still third party, something that did not belong to you. So at the end, who can guarantee that they were not going to reveal it, or someone hack the company and reveal all this information? There are a lot of stories I can give you about importance of intelligent genomic analyses, and the application are really limited by your imagination. This is another work from ETH where they store data inside synthetic DNA. So this is built out of synthetic DNA, this little bunny. So you can sequence this bunny again, send it to the sequencing machine, and then start reading the information. They even store a YouTube movie inside this bunny. The nice thing about this bunny, you don't need power to operate it, right? You can store the data for thousands of years, right? It won't be affected as long as you keep the bunny in a safe place. So it's a real nice thing that you can do with your DNA. This is another personalized shopping experience. We're not advertising the company, but this is just to give you more information about startup that can benefit from your DNA. This is a watch. Whenever you go to the supermarket, you can scan the barcode of the items and tells you if this is good for you or not, based on your DNA. If you have allergy to something, Okay, let's get done from all the examples. I can speak about those examples for hours, but let's move to the actual job. Now, the question we have, how and where to enable fast, accurate, cheap, privacy-preserving, and exabyte scale analysis of genomic data. Hopefully, for each of these terms or keyword, you got the motivation why we really need to care about all of these. And that is equal, all of this equal to intelligent genomic analyses. Do you have any questions so far? Yeah, feel free to interrupt, ask questions whenever you feel you have something not clear. Now, how we analyze the genome. So you got the blood sample or anything from your body, could be scanned tissues, and then you would like to get the full 3.2 billion characters of your DNA. It's not that easy. Unfortunately, we don't have, until today, any machine that can give you the full sequence of your DNA. Why is that? Please read our paper. So we describe a lot of things that restricting us from reading the full DNA as one piece. But it's all related to the technology. So far, we don't have any technology that go at this scale and read the chromosome. What we can do is old school fashion. So if you ask your grandmother about those tangled yarn balls that you would like to untangle them, what she would do, what any grandmother would do. So if you have this really tangled in a complex way that you cannot really solve, so the easiest way is just to cut these tangled yarn, right? And then try to solve the entire ball. And that's exactly what we do with your DNA. So we cut it into pieces, and then we give it to the sequencing machine, and now sequencing machine can read these pieces in much easier way. Currently, we are doing really good in terms of high throughput. We are providing this much of throughput in this much of time with this much of cost with one of the machines, which is Novo 6000. However, we still get small randomized fragments of your DNA. So it's no longer one piece, and that is the challenge. And for that, just to summarize the story, we have a sample collection. We got the DNA sample. We do what we call library preparation. This is done in the wet lab. You don't see normally these, a process. You got the fragments already processed, and then you feed them to the sequencing machine. The sequencing machine provides you raw sequencing data, and those raw sequencing data could be pictures, movies, or electrical signal, amplitude. It depends on the sequencing technologies. So far, we have 3 prominent sequencing technology that I'm going to describe. After you get the raw sequencing data, your job as a computer scientist or computer architect starts. So here we start only the computational steps and everything before done in the wet lab where you don't see normally. These are the 3 prominent companies. Oxford Nanopore, they are well known for portable devices. You can see they are very small, these handheld devices. PacBio, they are well known for a bit long and accurate reads. Illumina, the cheapest technology so far. Cheap and provide very short sequences, but very accurate. There are a lot of properties about the price and so on. The slides are already online in the course webpage, so feel free to check these properties. As I said, 3 different sequencing technology and 3 different outputs. So Illumina give you pictures, and any of these pixels in the picture telling you that this is, for example, if this picture for A, then any light in the A telling you that there is A here, 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 and so on. This is C. You have 4 pictures for one of the technology or 2. It depends on what technology you are using. But if you are using 4, then a picture for A, picture for C, picture for G and T. I'm going to explain the process in the next slide. O and T give you electrical signal, amplitude readings. So you got the amplitude this way, this way, this way, and you need to translate the change in the phase into A or C or G or T. For PacBio, it's similar to Illumina, but give you a movie. Okay, how Illumina sequencing works. So you have what we call a flow cell which is a glass surface. You already process the DNA strand, so you don't know the content of this in advance. And then you stick it to the glass surface. And then what you do from biology class, you already know that A should bind with T. You can think about it as complement. And G should bind with C, or vice versa, right? But you cannot have A binded with G. Using this information, the technology exploited this, and they start adding chemical molecules to the surface. So they add T, and then they wait a bit, and they have an optical sensor, and then they observe if there is a chemical reaction. If there is a chemical reaction happening, it means there was an A there, because I know I added T myself, right? Then the sequencing machine going to provide A, because I already know that I added T, and there is no other way to bind with anything except A. The good thing that I don't do this one by one, but I do it at very large scale. So I do it for all these strands at once. That's why I call this technology high throughput sequencing technology. Now I repeat the same thing. I wash the surface, and then I add A, wait for chemical reaction, take a picture, wash the surface, add C, wash the surface, add G. Because I don't know what is there. It could be a C, right? If I add T, nothing will happen. Then I need to repeat with these 4 molecules every time I sequence a single base. And that's what we got from Illumina machine. There's a nice video here. You can watch it on your own. I recommend everyone to have a look. Now, another sequencing technology. This is Nanopore sequencing technology. So how it works, we have this nano hole, and we let the DNA strand, after process it in the wet lab with the lipo preparation, we let an electrical current passing through the hole. We already know the amplitude of this electrical current passing here, and the DNA strand will affect the phase or the amplitude of the electrical current. So it will add a noise to it, it will add distortion to the amplitude. And then I'm going to read the new electrical current that already passed through the DNA strand. This is moving by itself due to the gravity, okay? So I keep reading everything over there. And this is what I get, basically, the amplitude, and then I use neural networks to convert those amplitude readings into ACGT. There's another nice video you can watch. This is the ONT device itself. This is the sequencing machine. It's really small, handheld, exactly same size of your hand, connected with USB cable to the laptop, and this is what we call the flow cell. I have here a few examples of the flow cell. I'm going to distribute it. If you can pass it to the others. Please have a look, give it to your friends. What you can see in the back, you'll see a sensor. There's a sensor in the back of the flow cell where it reads the electrical current readings. So the electrical current passes through DNA. The DNA will be inserted here in the flow cell, and then the sensor in the back will read these electrical current readings and send it to the UBS. And then we do some translation from raw signal into ACGT data. Now, this flow cell that you have in your hand costs $900 just to get that piece. And this is something that you throw away once you do the analysis. You do the analysis once or a few times, it depends on what you are targeting, and then you throw it away. However, this machine costs $1,000, where you stick the flow cell to it, and that is you can keep, because the nanopores, you cannot reuse them after some time. So you need to throw it away. It's a really expensive process. This is another paper summarizing the machine learning algorithms can be used to process the raw signal of nanopore devices. So if you're interested in this domain, please check this paper. And we have a nice work. One of them we already published, and few of them ongoing research on how we improve the raw signal of the nanopore, where we can do computation over there. Now, something common, all these technologies and all these devices, which they provide you pieces of your DNA and not the full DNA. And for that, what we need is really to solve it as if it is a puzzle. So you got the full picture, which is a reference genome. You got your pieces from the sequencing machine, and your goal is to stick them together based on this reference, and then you build your own DNA. Now, the three prominent sequencing technology, they do not provide you the full genome, but they provide you pieces, and those pieces could be small or large. Now, which one is better for you? To use very small pieces to solve the puzzle or very large pieces? The good thing about the small, although they are small, but they are very accurate. 99.9999 accurate. The long one, they are long, so they save your life. They help you a lot to figure out solving the puzzle very quickly, but they are inaccurate. So the accuracy could be somewhere between 5% to 15%. So you might get a portion of your DNA really different than what you have. How we solve that? By generating more data. So although it's noisy, but we require more data so that we overlap them. And then based, for example, on majority voting, we said, okay, probably at this location you have those and the rest are noise, just delete them. So you will pay more because you are going to consume more flow cells, the same piece you have in your hand. So Illumina is very short, but very accurate. Long reads or ONT, very long, but less accurate. And the hi-fi somewhere between. So the hi-fi, for example, 20 to 30,000. These numbers are a bit old. So the current technology is 20 to 30,000 long pieces. And the accuracy is really impressive, something close to the Illumina data. All right, so as I told you, the sequencing and generating the raw sequencing data is the last step before we start computational steps, right? So the sequencing technology is going to affect whatever algorithm you are going to develop, because the algorithm that operate over pictures is different than the algorithm operate over raw electrical signal. And this is our work describing these differences between the sequencing technology and what kind of algorithm you need to build for each of those sequencing technologies. So I recommend everyone to read it. Okay, looking forward, will we get a sequencing technology that can read the entire genome as one piece? That is still an open question, and we discuss it in our paper. Now let's talk about the barriers to enable intelligent genome analysis. Yeah, feel free to stop me and ask questions. I don't know, the clicker is not working, so I have to move back and forth. But the first significant barrier, the sequencing machine that we were explaining, or the data generator, basically, is much faster than any general purpose computer that we are using. It's really high throughput, such as providing you a huge amount of data in short time. And until now, we are still using those computers to do the analysis. But this device can do only one job, which is sequencing. That's why it's really specialized to the job. And we would check the performance. Any sequencing machine, or decent one basically, can analyze 48 human genome in about 2 days. And to analyze that, we need 32 CPU hours to analyze just one human genome. So there is a huge gap between the performance of sequencing machine and the computer used to analyze the data. The second bottleneck or the barrier is expensive data movement. So I already tell you that there's 3.2 billion characters need to be moved from the sequencing machine to the storage or the general purpose computer. And then you move the data from the storage, you fetch it to the main memory, to the CPU or the caches. And this includes a lot of trips, round trips from the compute systems. And that actually accounting for about 40 to 60% of the entire system energy, which is really huge. That is still excluding the computation. So still you're not doing any kind of computation. You're just moving the data for the analysis. So data analysis is performed far away from the data or where we store the data. The third barrier is that we are neglecting metadata, which is the data that define the technology, the sequencing technology that we are using. If you don't care about what is the input to your problem or what properties this input has, then you are losing a lot of features that you can exploit. For example, if you know that Illumina data is very accurate and it's very short, then the chance to have a variation is very low. Why is that? Because you have very long human genome and the variation happening at certain location where you know at this chromosome, at this location, you will have a disease, right? So it makes more sense to start doing exact matching to these short pieces since they are very short and very accurate. So you don't need to consider errors, for example, within these pieces. So by knowing more about the data you are processing, you could exploit this information and do very efficient algorithm. And I'm going to talk about GenStore, which is one of the work that we exploited this information and we do with the processing in storage. There are many other barriers that are very specific to each analysis step that we do. And there are really large number of computational steps that you can target to process the raw sequencing data, which is images, electrical signal, or movies that you need to base call. And then you got the read set, you got the reference genome from somewhere in the cloud. They are already publicly available for free. You can download them. And then you start doing indexing, read correction to the read to remove some errors, then read mapping, where you locate each piece you got in the read set to the reference genome. So you have very long reference genome, and you want to locate every read where it's located in the reference genome. After read mapping, you can do many other applications. Taxonomic profiling, for example. So you can specify what virus exists in the sample, what bacteria exist over there. You can do assembly even. Assembly is when you don't have a reference genome for the analysis. You don't know in advance what you are targeting. So you want to build or overlap these reads together and build a graph on top of it. Then you can solve the bubbles, any rounds in the graph, so such that you have very smooth linear graph. And then in this way, you can get your DNA. You don't need a reference genome to compare the read to the reference genome. I'm going to explain more about read mapping in the few next slide. As I said, there are some barriers that are very specific to each step in the pipeline, starting from base calling, error correction, read mapping, variant calling, and metagenomic profiling. And there are a lot of challenges over there. In our paper, the recent paper I assigned to you for reading, so we profiled the entire pipeline with different steps. This is the sequencing from Illumina, sequencing from ONT, sequencing from PacBio. And as I said, sequencing technology dictates the computational steps. So you cannot use this base calling step for this sequencing technology. All of these steps are really customized, specialized to the sequencing technology. And if you check the throughput of each of these steps, you will see sometimes the base calling is the bottleneck, sometimes the read mapping, sometimes variant calling. It depends on the sequencing technology. For ONT, which is the portable devices, base calling is really expensive. You can see the throughput is very low compared to the rest of the pipeline. Variant calling is also another expensive step. But what is common in all of them, that read mapping is always expensive. That's why we're going to focus in the next part of the lecture on read mapping. If you have any question, feel free to ask. If you want to take your time looking at the picture. Okay. So for that, what we envision as an efficient computer architect or an experienced computer architect or computer scientist, as all of you, you would like really to target the entire compute stack. You cannot just optimize the data and that's it. You really need to build efficient algorithm customized to the problem you are trying to solve. You need to build architecture even using Fpga logic, for example, or GPU to accelerate the processes. So you need to work in the complete stack. Of course, you can ignore the device level or electron levels since this requires fabrication facilities, which normally we don't have at universities. To motivate you further to target the complete stack, this is a simple problem of matrix multiplication. You have a matrix, you want to multiply it with another, and you get the results. And hopefully all of you know that it requires 3 nested for loop, right? To traverse through the 2 matrices and try to solve that. Now, If you use different programming languages, this is the run time, and this is the speed up you could achieve. If you start with Python, which is kind of really easy, is like a human readable code, right? So you can read it and easy to code, but it's not efficient. But if you go to C, you got kind of 50x of speed up. There is no special optimization yet, just using different programming languages. Now, if you move to parallel loops, for example, OpenCL or any kind of tool that enable you to add the pragmas to the for loop to unroll it and make the things faster, or if you move to AVX acceleration, SIMD acceleration, single instruction, multiple data, then you can achieve a huge speed up by just moving into lower level programming language. That's why we always encourage everyone working in bioinformatics to use C programming language. To motivate you further, this is another benchmark available in this link. You can check it out. That is just to read the read set you get from the sequencing machine, FastQ parsing. Just reading the genomic data you got from the sequencing machine. If you use Rust, it's the most efficient one in reading the plain text. This is the compressed text. This is plain. C is maybe second best, but Python is the last on the list, right? So don't use Python for that. Always use C. Hopefully that motivates you a bit on the efficiency or the effect of using different programming languages, and that is not to mention the hardware acceleration behind it. So we really need intelligent algorithms and intelligent architecture that handle data well. Why is that? Because if you don't know what you are targeting as hardware, you are going to miss a lot of nice things that you can exploit. I'm going to show you a story about a gatekeeper, for example. We designed an algorithm for Fpga, but given that in mind that we know what the Fpga we are targeting, making the size of lookup table available to us, then we could design very efficient algorithm to exploit those sizes of lookup table such that we reduce the resource utilization. Now, let's explain more about read mapping, which is the problem of this, solving the puzzle. You have the reference picture, you have the pieces, you want to solve this puzzle. So the first step, you go to this link, you download what we call reference genome. This is 3.2 billion character representing reference genome for someone else. It's not you, of course. This is a collection of people where we build the reference genome for them, and then we use it everywhere in the entire world. Everyone in the world working in this area, they should deal with this reference genome. You can see some properties, but this is publicly available from NIH. Now, on the other side, the things I was showing here is the reference picture, the complete picture. Now, you need the pieces. Either you do the sequencing using the sequencing machine you have in hand, or you could download it also from internet. So following this link, go to SRA database, you can download it directly, and you get this size. This is the file if you click here. Now you got the scattered pieces and you got the full picture. Now your goal is to assign each piece to a location in the reference picture. Let's learn how to map read. Now a brute force algorithm would be basically to pick one piece from the read and traverse through the entire reference genome and try to find the best location over there. So you just pick the piece, check every location. Is it really good here? Does it match to this location? How many differences over there between this piece and that piece, and so on? And you pick the location that has the minimum differences between the blue piece and the red piece. However, this is really expensive. The time complexity is really huge for that. And that's why we call it brute force, because you are checking every single location. In real life, what we do, this is the file you get, the reference genome. This is the read set. This only line, the second line, is the read itself. This one is identifier. This is just a sign to split these two lines from each other. And this is in ASCII code, the quality of this base. And that is F, the quality of A, and so on. So the sequencing machine gives you the quality for each base. So it's going to tell you that I was not confident whether this is A or T, if this quality is very low. So you can leverage this information and ignore the bases that has low quality, for example, in the FastQ file. All right, going back to read mapping, how we do it. Since this is a huge workload, we cannot search it linearly. We cannot traverse through it. So we are going to collect some pieces, some subsequences, and store it in a hash table or any kind of indexing. So we build a hash table, then it's easy. In O1, you can query the hash table. Do you have this piece? No. Okay, move on to the next piece. Do you have the other piece? Yes. Then collect these pieces. Save them in the hash table. Once we generate the hash table, we go back to the read. Now collect pieces from the read sequence the same way we collect from there, but don't store them in a hash table. Use the first piece to query the hash table and find the blue pieces. So we find here and there. Now do the same for the green piece. Collect the green piece, try to find the location here and there. The red, same thing. Now, what is next? We already defined the location of each of these subsequences. What we should do next? Now we said since the blue is followed by green, followed by red in the red sequence, so to make more sense to try to find a location or a region in the reference genome where we exactly have the same order as in here. So we have the blue, green, and red, right? Which is basically this region. So it sounds this region is similar to the read, then okay, let's try to investigate further the differences between this line and this line. And this is how we do it in real life. Now, we convert the reference genome into kind of indexing data structure. Normally, we use hash table. So hash table is the most popular indexing technique for all read mapping algorithm. Any read mapping algorithm you can check, starting from 30 years or so, they should use kind of hash table. There are a few other algorithms that use other techniques like Boris-Wheeler transformation or suffix array, for example. There are many more, R-index and so on. But the hash table is still the most popular one. This is how it looks like. So this is where we store the hash value of the seed, not the seed itself. So we store the hash value of the seed picked from the reference genome and the location, because we need the location to retrieve where we are going to pick the read and put it in the reference picture. So that's why we store the location of all these pieces. Now we call this subsequence or a k-mer or a seed, where k is the length of this. If you pick 15 characters from the reference genome, then we call it 15-mer, and so on. Okay, now just more information about the index size, how large it could be. So there are different read mapper tools, and this is different index size. Why they are different? Why we got 16 gig, 7 gig, and 4 gig? Although all of them, they build a hash table for the same human genome, which is same size. Why we got differences in hash table? One of the main reasons is the way we pick these subsequences, how we pick the k-mers. So there are really tremendous amount of algorithms that can be used to fetch these seeds. So they could be overlapping to each other. Non-overlap, you have a question? Okay. Yeah, feel free to stop me and ask questions. I will be happy for that. There are different ways so that you can pick every overlapping seeds. So at each location, you pick the next 15 characters, and then the next location, another 15 characters, another 15 characters, and so on. That will give you a huge hash table because you are picking almost every seed, and that will be 15 times the size of the human genome, right? Because at each base, you pick 15 characters. And then the next base, you pick 15 characters. But there are also other ways. For example, blend from our group is where you pick less number of seeds, stop-mers, sync-mers, adjacent, non-adjacent, space seed, and so on. All of those are already published work. So I'll be happy to discuss them if you need more information. Now, using different indexing methods going to affect the entire performance. So if you use a hash table, the CPU time sometimes will be faster, sometimes slow. It really depends on the way you pick the seeds. There are other methods called Boris-Wheeler transformation using the FM index, which is a compressed fashion of the genome itself. So it's always fast because you are going to reduce the amount of data and you are not storing everything. You are storing just small information about the seed itself rather than the entire seed. And the memory is low because it's compressed representation of the genome. The hash table in terms of memory can be large or small. It really depends on the way you pick the seeds. Again, there are other suffix array and other implementation, and this is still open research, open problem research. It's not solved yet. Every few years, we got a new data structure to solve that. Now, assume you already queried the seeds you got from the read. You already know their location in the reference genome by checking the hash table every time, and you know what region you are going to target. What we do next is trying to sort the location we got from the hash table so that we got the nearby location in the reference genome. And then if you got a lot of nearby location in the reference genome, then we said this is the region that we are targeting. What is next? Since here is not necessarily to pick the seeds that are next to each other. They could be really apart from each other. So for example, you pick just 3 seeds from very long sequence. In this way, there are gaps between every 2 seeds, right? And that's why we need now fine-grained evaluation or comparison for the 2 sequences. And we use for that dynamic programming to check every base, how it compares with the other base from the other sequence. So this is the read itself, the complete read. And this is the complete region that we fetch from the reference genome based on the indexing method that we use. What is dynamic programming? So one of the way to solve or to use dynamic programming for is edit distance. So I was telling you in the beginning about Hamming distance, but edit distance is more richer. So it's enriching the comparison with more insertion and deletion. In Hamming distance, you could only figure out the differences or the matches between 2 sequences. In edit distance, you can detect also insertion and deletion. How is that possible? Let's see an example. I want to compare organization and operation. So you can use dynamic programming, which is the table where you fill with information, and then you can reuse every time you want to build new row. Now, after building that, you may get a solution with this arrangement or a solution with that arrangement. This is how you arrange the 2 characters or the 2 words so that it looks similar to each other. So O matching O here in insertion means ignore, this deletion, this matching, this is insertion, this is deletion, and vice versa. It depends on if you read it from this or this line. And those exactly matching to each other. Now, this is another way to do it, where you can match the characters so that they are different. Or similar, sorry, not different. We always would like to make them different, and what number of operation that we need to do to make them similar. And we pick the arrangement that provide the least number of operation. For example, here another one, organization and translation. So there are 3 different ways to arrange these characters. And now which one provide you the least number of operations? So you can see here 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Here, just 4 arrangement to make them similar. Then you said, okay, let's pick this arrangement and save it somewhere. And that's going to tell you that the region you pick from the reference genome and the read sequence, they have 4 differences to each other. For this problem, we always use dynamic programming. Why is that? Because when you compare the first character to all possible location, you already compute that. Now, when you move to the second character, you don't need to compute the first and second together because that is already computed. And this is how we store the results in this line. Now, since already stored over there, when we move to the second character, you just read the previous value and you do further computation to compute this. You read this value, you compute that. You read this value, you compute this, and so on. So when you reach here, you don't need to compute all these characters again. They're already stored here. This represents this and this and this, and so on. So you just need to compute this based on this value, this, and this. So you read every time 3 values to calculate any single cell in the table. That's why we call it dynamic programming. All right, so the most famous dynamic programming algorithm until now is Smith-Waterman, based on our paper. And the Hamming distance is the second most popular. Hamming distance is just XOR operation between 2 strings. So you have 2 strings, you just do XOR between them so that you can figure out the matches and the mismatches, but you cannot recognize deletion or insertions. Okay, so we were explaining how you do read mapping. Hopefully that set the stage for you, already introduced you to the topic. Another application, if you don't have the reference genome, and if you don't find it in the NIH or NCBI database, what you can do is to build the Novo genome assembly. What is it? So at each node, we have the read we get from the sequencing machine, and then we try to link it to the next read. So whenever this read shares something with the next read, then we link them with an edge. So you have these nodes, the read, and this is the edge representing the common part between the two reads. And then we keep doing that until we build the entire graph. Now, since we only consider a small part of the read for the overlap between the two nodes, and also the reads, they have their own errors because the sequencing machine is not perfect, right? They introduce their own error. So we need to solve this bubble. We got this kind of bubble, as you can see, is going back and forth in the graph itself. So those are very bad for us. We don't need them because our goal is to link all the reads together such that we build the entire linear representation of our genome, which is 3.2 billion characters, right? So we don't want to go back and forth within the graph. So it's really a difficult problem. It's not easy to solve the de novo genome assembly and can take a lot of resources to build the assembly for that. In real life, it would look like something like this. This is for an entire viral data, which is very small. If you go with the human genome data, something really huge. All right, so now as a computer architect, we want to solve something or accelerate it. What we should do? First, understanding what makes this problem slow, right? And for that, we are profiling one of the state-of-the-art read mapper called Minimap2. And then we find the different steps and how they contribute to the execution time of the entire pipeline. I was telling you that there is indexing, seeding, where we collect seeds from the read, and then pre-alignment filtering, and then the dynamic programming step. All these steps in one read mapper. And if we check, you'll see that about 45% plus 16%, they are using dynamic programming, and they are accounting for more than 60% of the entire execution time. So read mapping, if you want to accelerate it, you want to target the dynamic programming and start reducing the workload for that. If you check this paper, they provide you a theoretical proof that you cannot do better for accelerating sequence alignment if you want to keep the optimality for all use cases. So optimality and generality. What we also figure out, we observe that the time that you are calling dynamic programming to do the comparison between every read and the region in the genome, in the reference genome, they are dissimilar from each other. So you got the read sequence from the sequencing machine, you got the region from the reference genome where you detect using indexing, and now you want to compare them with dynamic programming, right? 98% of the time where you do this comparison, they are dissimilar from each other. They are totally different from each other. And that's why it's really a waste of time that we are calling an expensive algorithm while the sequences are different from each other. Now, how we improve that? You have two options. Either we continue the lecture or we take 5 minutes break. 5 minutes break. Okay, let's go for it. Yeah, we'll be back after the break. Yes.
In reference to Homo sapiens, it seems that at least the starting of the gene are usually several Is it because the telomere sequence are very like
Because two things. This region is very complex in the chromosome, plus the sequencing machine cannot read it. So to provide N means unknown, something really unknown. So we don't know what is it.
This basically corresponds to the queuing.
Yes, yes. And this is the exactly what you see from the beginning of the file. So once you fetch like the first few lines, you'll see exactly this content. That's why I kept it over there. So once you see it, you don't get scared. What went wrong? Why it's not ACGT data?
That means like. So I also see like asking.
No, this is the ID.
Oh no.
Should be N or ACGT, nothing more. Only these 5 characters. It's basically 4, but the 5th is unknown. The first line is only identifier, unique identifier, to know what you are reading, right? If there's a viral, you'll see the name of the viral plus accession number, which is a unique identifier to the species. Yeah. Why?
Because we were showing some colors.
Yeah, this is nice. Diversity always perfect.
Maybe we shouldn't see any colors.
No, you know, African people always the best in the world because they have more DNA. You see the opposite story.
The more DNA is better.
Yes.
Onion is better than you.
Good one.
It's more useful, I guess.
Hey, Julian. How many of you from PNS class? 1, 2, 3. Okay, bunch in the middle. Oh, yeah. Oh, I see. Maybe send us email and we will check.
So there should be a
It should be. Yeah, but send us email. I will give you the exact instruction. No, send us to. I don't want to play with this, but if you go to the course webpage, you'll see in the home mailing list where you can send to all instructor of the course. So there is an email before the list of TAs in the first page of the course page. Yeah, send to that email, and before just right before the table. Okay.
The concept, just want to make sure they are basically when we take from whatever we got and we want to compare it to the human one, for example. Only basically check chunks from the original one, map it and then go to the next 2D one. The other one also check some support, then trying to see if there is a mapping. But the thing is, when we do the hashing, the hashing take account, but small difference is not a problem, basically.
Oh, that's a great question. Yeah, this is basically one of the limitations of using hash table because you require the next 15 characters to be exactly the same.
Yeah, and that doesn't make sense.
So if there is one difference in there, you will lose it. But the nice thing that we pick more than one piece. So if you miss some of them because there were variation in the middle, the other pieces you pick from the read should be matched with the hash table.
Okay.
Because you have the read, could be million long, right? It's not just 100. So from million, if you pick every 15 as a seed, then you pick maybe 200 seeds, right? Or something. If you lose half of them, you still have the other half.
Okay.
If half of them don't match with the hash table, you still have other half that match with the hash table. Then you're still in good shape, right? But there are also other methods that try to solve that. For example, instead of giving a chunk of bases as they are to hash them, you pick like every other character. One character, leave one, one character, leave one, and then hash these 15 characters. So you are taking 30 long region, not 15, but you hash only 15 of them, and you leave other 15 for errors. So you can tolerate more errors. There are different techniques to solve this issue, but this is a very great question. Yeah. No, you cannot. Yeah, that's fine. Yeah, please watch the online lecture.
Exactly. Question regarding constellation. You said you would send concerning how I can access the classroom.
Yes.
So that was.
I saw your email, but I'm going to do it after the lecture. I'll send you the GLIP group and the instruction. No worries. You'll get it today. Yeah. All right, guys. So the 5 minutes is already over. It's more than 5, actually. Okay. So we already profiled the complete pipeline for read mapping, and we figured out that sequence alignment is the most expensive part. So that's why we have this work where we try to leverage all the existing information or algorithms to solve this problem. And we have a lot of works in Safari Research Group at ETH where we try to address similar problem. So we target different part of the compute stack or compute system, including the processor, the DRAM, storage, and the sequencing machine itself. So those are the work, try to accelerate different part of the read mapper. Some of them, we do it inside the SSD system, some of them inside the DRAM. And during the next few weeks, you will get introduced to some of these topics from the author themselves. So the first author going to present some of this work in detail and in depth. All right, so the first ongoing direction to solve this problem, which is being, as a read mapper, very expensive, how we can accelerate the things, is what we call seed filtering technique. And we got the question in the break about that, how we can accelerate that or tolerate more errors, basically, because we pick 15 characters as they are, as one chunk, one block, and we save them in the hash table. Now, what if there are a difference in the middle of this seed? Then how we are going to match it with the hash table? You'll never get a hit, right? You always get a mess. The second direction are what we call pre-alignment filtering. And that's going to filter the data that we send to the dynamic programming algorithm. So you have the dynamic programming to check the similarity between two sequences. We're going to filter out the data before we send it to the dynamic programming, and then only very similar sequences, we send them to the sequence alignment. The third direction is to accelerate read alignment itself, which is sequence alignment, or the dynamic programming algorithm. There are ways to accelerate it. If you don't want to accelerate the algorithm itself, at least you can use parallelism, where you have multiple processing elements. Each of them can solve the same problem for different sequences. That is using FPGA or GPU or specialized architecture. Let's go in depth in the first direction. This is what we call fast hash from our group. And what we do, we solve two problems. First, adjacent k-mers and cheap k-mers. In adjacent k-mers, we said, it makes more sense to pick the region that has seeds adjacent to each other rather than picking a region that has seeds very apart from each other. So let's have an example. We have a read, we have the reference genome. Let's assume that we pick 3 seeds from this read. So we pick the red one, the green, or maybe the yellow, but it's kind of brown. And then the third one is the green, right? So kind of we pick 3 seeds from the read sequence. Now we want to query the hash table, and the hash table will give you the location. So let's assume these are the locations. The arrows this is the location in the reference genome, another location, and so on. Now, if you look by naked eye, just at the reference genome and the location that we got assigned, you'll see the three arrows, the red, yellow, and green. For example, they are scattered here. So this is potentially one of the region, right? But this is maybe another region where it's flipped. So you got the yellow before the red, and so on. So you can make decision based on the location only, and you can build your own filtering algorithm based on your target. For example, if you are targeting only small variations and snaps, so it makes more sense to pick this region rather than the other regions. Why is that? Because these three seeds are next to each other, or what we call adjacent seeds, right? But if we are targeting structural variation, so we are targeting very large insertion or deletion, so it makes more sense to target this region, right? Although the green here is coming before the yellow, but there are some inversion happening as part of structural variation. So it could be insertion, deletion, duplication, or inversion. The region could be inverted in the genome. So it depends on what you are targeting, what kind of problem you're trying to solve in variant calling or in variation detection. You can design your own algorithm to pick these regions in the reference genome. Now, going to the second observation or the second work. So we said this is a valid mapping, but as I said, there is no one standard solution. It really depends on what you are targeting. Now, going to the second observation, which we call cheap k-mers. What do we mean by that? So what is expensive k-mer first? We define expensive k-mer as the k-mer that got a lot of locations from the hash table. So once we query a seed in the hash table, we got like 10 million locations. This we call it expensive because we are going to check 10 million locations in the reference genome and see whether they are good or bad. And we are always looking for one or a few locations in the reference genome. So we consider this as a noise. Let's ignore it. Let's take an example. We have a read sequence. We set our threshold to 500. What's this threshold? Now we will see what is it. We query the first sequence or the seed we got from the reference genome. We got these list of locations. So we got 4 locations. We got another seed from the read. And we query, we got this location. We got another seed from the read. We query, we got 1,000 location, 2,000, 1,000, and so on. Now, which one is expensive? Which one is cheap? There is no standard here again. It really depends on you and your application. But since we set the threshold to 500, then we said anything above 500 is considered expensive. And anything equal to 500 or less is considered cheap. So let's ignore the expensive ones, which are highlighted in red. If you ignore them, which we consider them as a noise, not useful information, and then we consider only the blue ones. So these are the blue ones. And previously we have 3,000 locations to be checked, but now we have only 8 locations to be examined as part of the reference genome. Now those are very cheap to compute, right? Instead of doing 1,000 locations where we need also to sort them and then do some kind of computation on them. So sorting always expensive. And if you reduce the workload for sorting algorithm, you are going to have a faster operation over there. That was fast hash. At the end, you can achieve up to 19x of speed up just because of those 2 techniques. Again, this is from our group. So as of all our papers, all of our work from the group, they are always open source. So you can go to the GitHub of Safari research group and download them, play with the code. It's already in C. So you can do many nice things over there. All right, let's go with the second direction, pre-alignment filtering. So what do we mean by pre-alignment filtering? As the name itself can tell everything. So we have a step that operates just before the alignment step, which is the dynamic programming. And what we are trying to solve. We observe that always calling the dynamic programming algorithm is always expensive because it's a quadratic time algorithm. We need 2 nested for loop to solve this matrix or the dynamic programming table. So it's really expensive to traverse through the entire table. And our goal is to reduce the need for dynamic programming algorithm. We said it might be safe to not touch the dynamic programming, keep it as is, but reduce the number of times that we are calling this expensive algorithm. How we do that? We observe something in all genomic strings. So when you have genomic strings, either they are similar to each other or dissimilar to each other. Now, if they are similar, we keep them. If they are dissimilar, they are expensive. Let's remove them. And we ignore them. We don't need them. And again, how we define dissimilar or similar, we should use a threshold. So if there are 5 differences and our threshold is 6, then those are similar sequences. Let's keep them. If they are similar, we need to find the number, the type, and the location of the differences. So every difference between the 2 sequences, we need to find the number, the type, and the location. Why is that? Because it's really important to know where's this variation happening, at what location in your chromosome in your genome, right? Because after that, we are going to say the disease is happening because of that variation over there. And we need to know the type, whether it's insertion, deletion, inversion, duplication, and so on. And how many of them they are happening. So we said, okay, let's not call the sequence alignment all the time. Let's have a filter that check the similarity between the 2 sequences in using heuristics. They are not need to be perfect all the time, but at least they are perfect in some time. That is the definition of heuristics, right? But we also have some 3 main properties that we need to keep. The first one, we need to filter out most of dissimilar sequences. So we need to have the ability to tell quickly if 2 sequences have a lot of differences or not. And we need to filter out only the ones that have a lot of dissimilar or differences. Now, why I said most of and not all of them? Because if I'm able to detect all the dissimilar sequences, then I'm perfect algorithm, right? And if you are a perfect algorithm, then you are probably doing same job as sequence align. Then I'm not gaining anything. Now, the second one, we need to preserve all correct mapping, and that is all here. Why all? You can put maybe 1,000 thread lines here. Why all correct ones? We need to keep them. Because if we lose some correct sequences, then it might affect the discovery at the end of the variation, right? I may not discover the variation because I just delete some correct sequences that should map to the reference genome. That's why I need to keep all of them. So it's okay to filter 80% of the bad guys, but we need to keep 100% of the good guys. The third property, we need to do it quickly because this is the main purpose of pre-alignment filtering. We need to perform much faster than sequence alignment. And after we introduce this principle, which we call pre-alignment filtering, we start proposing some algorithm to solve the problem. Let's jump quickly to the example because I always like examples, much better than text. Think about 2 sentences or 2 words that you would like to compare them. The first one is the read. The second one is the genomic region you get from the human genome. Now, Istanbul and Istanbul. If you want to compare them directly using Hamming distance or cheap computation, you got directly an exact match. So that is 8 matches and 0 mismatches, right? Now, I want to do something to the text itself, assuming that there was an insertion or deletion or some alteration or variation in the genome. So let's delete the character A. Now, it will be a new word, something like that, right? So now I just delete the A character. I didn't do anything else. Now let's compare them. If I do exact match between the 2 words, now I got 3 matches, but 5 mismatches, although I did only single change. Now, how to fix that issue? We said, let's have another copy of this guy. So we add another copy, but we shifted 1 step to the right. Now we have 2 copies of the same read sequence, and we have 1 region from the reference genome. Now let's do Hamming distance. So we do with the first and first, we got 3 matches. We do with the first and the second, we got 4 matches. Any question? Hopefully it's straightforward. So you got 3 and 4. If you add them up using XOR and add operation, you will get 3 zeros and 4 zeros corresponding to the matches that we got. And what you get, 1 representing the single change. And what is also nicer, that you got the 1 in the same location where you delete the A. So you see 3 matches and then 1 change in the exact same location where you delete the character. So this kind of very cheap computation can tell you quickly how many changes between the 2 sequences and what is the location of the change. So this kind of computation, very cheap to do in Fpga, for example, right? Just XOR between this, and then shift for this, and then XOR, and then AND operation where you got the result. And then just counter, simple counter, will tell you how many variations here. So you can see how simple algorithm can solve a huge problem in read mapping or in genomics. Now let's see a real example as well with longer sequences. So this is the query, this is the reference. The query is read, and the target is a reference. So either you can use query and target or read and reference. And what we do here, since we don't know how many changes, in the previous example, we did the change ourselves. We delete the character ourselves. But in real life, you don't know what are the changes and what they are and how many, right? So for that, we need always to build 2E plus 1 of these copies. So in the previous example, we built only 1 copy. So we have the original text and a copy of it that is shifted to the right. Now we need to build here 2E plus 1. What is E? Edit distance threshold. So it's up to you. How many errors? You want to have 4. If you want to have 4, 3, then E is 3. Okay? So you need to build, in this example, 7, because 2E plus 1, which is 2 times 3 plus 1, 7 vectors. Now what they are? The first one is just Hamming mask, the XOR between the 2 strings, such that you got this, right? You can see if you compare this, exactly same. So you got 0, 0, 0. But here it's not the same. This is T and this is G. So you got 1. And you continue doing the same thing. Now those, we call them 1 deletion mask, 2 deletion mask, 3 deletion mask. What they are, it's when you shift the query 1 step to the right and then do XOR with the reference. So you got this one. So you shift this 1 step to the right, you compare with this, you get this. Now for the second one, you shift this 2 steps to the right, then you compare with the reference, then you got this. You shift 3 steps, you compare with this, you get another one. Now for the insertion, you do the opposite. You shift, but in the opposite direction. So if you were shifting to the right, now you will shift to the left. And then you compare, then you will get this direction, and so on. Now why we shift in both direction, right and left? Why we shift in both direction? Excellent. Because we don't know what type of insertion or variation we will have. So we may have insertion, may have a deletion. Now, if we insert a character in the middle, all the trailing character will be shifted to the right, right? Same as in Istanbul. In Istanbul, we did, if you remember, we delete 1 character, then all these 4 characters are shifted to the left. Now, if we insert 1 character here, then all these will be shifted to the right, right? So that's why, based on the type of the insertion or the deletion, the type of the inversion or the variation, all those shifting will be done to the right or to the left. That's why we need to consider all cases as shifted to the right or shifted to the left. Okay, so hopefully it's clear how we build the 2E plus 1 masks. Now what we do next, since as we did in the previous example, we did XOR, right? And that is the result of the XOR. And then we want to do AND operation between the 2 sequences, exactly as we did in the previous example. So we did XOR to the 2 copies, or the 1 copy and the original one, and then we AND the results. Same thing here. So we already built these vectors using XOR and shifting, and now we want to AND the results of this. Let's look into the results. This is the result of ANDing all those vectors together. So whenever we have 0, it always dominate because we are using AND operation, and then the zeros will appear directly in the end results. Whenever zeros dominate, 0 dominates, and so on. Here, we have zeros here, and we have 0 there. So they will dominate these ones and appear there. Now, if we compare this to sequence alignment. So as I said, this is not sequence alignment. This is heuristic algorithm, very cheap algorithm to do the comparison between two sequences. Now, if we see sequence alignment, this is the results of, this is the results of sequence alignment. This is a perfect algorithm, an exact algorithm. So you will see there is variation, variation, and 2 variations over there. So in total, there are 4 variations. But in our method, we detect only 1, 2, and no more here. So we detect only 2 errors rather than 4. That is fine. Why? Because we didn't say there were 4 errors while there are 4. So we didn't overestimate the number. We underestimated. And when we underestimated, we said those number of errors are already less than the threshold. Then they are going to pass the filter, and they are going to be examined with the sequence alignment. So in this case, we are in safe side still, because we never filter them out. We just let them go, or passing the filter. And that's what we call false positive, because our threshold is 3 based on what we define here, 2e plus 1, right? Our threshold is 3, so we can afford only 3 errors. But this in reality has 4. So we shouldn't let it go or passing through the filter. And that's why we call it false positive. Now, one way to fix this is, instead of doing AND directly, is to reduce the number of zeros. So you can observe here that what caused this error to be disappearing is the zeros, right? Those, whenever you have a 0, dominate here, and then we delete it from the results. Now, one way to, or a heuristic to solve that, is to ignore any single 0 or 2 zeros. Whenever we have single 0 surrounded by 1 or 2 zeros surrounded by 1, just switch them to 1. You'll see this much cleaner version of the mask where we don't have any single 0 or 2 zeros, all more than 2 zeros, 3, 3, 3, here a bunch of more zeros and so on. Now we play with the data itself, right? We change the representation of the data. That is fine. Why is that? Because we are going to increase the accuracy, but in the same time, we are not causing any correct one to be deleted. So now if you check the AND operation for this new copies of the mask, now when we do AND, we got this result. So how many ones? 1, 2, 3. So it's still going to be accepted, right? Because our threshold is 3, and anything equal to 3 or less will be accepted. So those 2 sequences will be accepted, although they should be rejected based on the perfect alignment. But at least we reduce the problem. It's no longer a big problem for us, since the accuracy for this sequences and the other sequences, it's already improved. Now, talking about how we switch the single 0, 2 zeros, why not 3 zeros? Why not 4 zeros? Why not 5 zeros? That is heuristic again. So you need to play it and try and error, right? So you sweep the number of zeros to be switched into ones and see what is the best performance for these algorithms. Now, going back to compare with the dynamic programming algorithm. So this is the dynamic programming, and this is what we built in Gatekeeper. So in here, if you remember, I told you this is dynamic programming, and this is the equation that we follow to fill the table. And if we calculate this, we need to check this, 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 and then after that, we decide what is the value of this. Based on this, you can see these are 4 different indices for the table, and this is the value that we are targeting, I and j, for example, this or this. And then we need to check this, 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 in order to fill one entry. However, in Gatekeeper, we don't need to depend on any of the previously calculated entries, because everything is independent of each other. You do XOR, for example. You could do XOR for the first character, also for the 100th character, right? At the same time, because we just XOR between the 2 sequences. Likewise, when you shift 1 copies to the right or to the left, so you could shift any part of the read independently from the first part of the read. So you could shift it alone and then do the XOR. So you really can divide the read into pieces and process each of these pieces separately, totally separately from each other. And then you can do AND operation, you get the results for each of these pieces and just collect the results from all of them and then add the sum to the final results. So it's really nicely parallelizable algorithm, and there is no dependencies at all in building this. So anything can be computed independent, and then you build everything in one shot, assuming everything fit into the lookup table or the FPGA. All right, and that is a nice thing for hardware acceleration. You could target FPGA, GPU, or SIMD acceleration with that. Now we built the first FPGA pre-alignment, FPGA-based realignment filter on that time. That was, I think, around 2015 when I was doing my PhD. And we used that to reduce the time or the number of times that we call dynamic programming. So instead of always sending the data after indexing to alignment, we use the filter to filter out most of the sequences that are dissimilar from each other to the sequence alignment. And now, how about the resources? How we are going to implement this in FPGA? So I think the most of the algorithm is very trivial, right? It's very easy to implement. You can check the C implementation of the code on the GitHub. It's really easy, straightforward. The most challenging one was how you replace single zero or two zeros into ones. When I say challenging, is challenging when you consider optimization, not when you consider implementation. You can do a bad job in implementing it, yeah, but you are losing the performance. So you have to do very efficient implementation, especially when we target FPGA. Every lookup table in the FPGA matters. So we said, let's use a number of lookup table, all of them operate in parallel, such that we do the processing for the entire mask at the same time in one shot, all of them in parallel. Now we said we need to decide on the size of the lookup table, how many input for the lookup table we need. Look at these two examples. So our goal is to replace any pattern like this or a pattern like that, or 1, 0, 1, and something else. Don't care, right? The X here, don't care. So in 1, 0, 1, it's trivial. You can find any pattern 1, 0, 1, and that's it, right? Which is need at least a 3-input lookup table. So you need to target FPGA that provide you a lookup table with at least 3 inputs. Now for this case, we need 5, not 4, not 6, not 7. We need 5 input lookup table. Why is that? What is common between this pattern and this pattern? Here, don't care, it means you can ignore. You don't care about the X. Here, same thing. So if you get 1 to the left or 1 to the right, that's good enough. But the problem with the other 2 bits, so either to the right, you should get 0, 1, or to the left, you should get 1, 0, right? To capture these 2 patterns. That's why you always have the bit you are targeting in the middle and 2 bits to the right and 2 bits to the left. That is in total 5 bits. In this way, you can cover this case, and you can cover also the case of 1, 0, 1, which is 3 bits. So you always need to check 2 bits to the right or 2 bits to the left in order to figure out whether you are targeting this pattern or this pattern. If you're targeting only 3, what if you got 0, 0, 1, right? Or if you got this pattern alone and you have 3 input lookup table, you cannot capture that one. What if 1, 0, 0, 0, and you have 3 input lookup table starting from here? Then you will see only this pattern, but you will not see the 1. That's why we need always 5 input lookup table. Yes. If you have 4, you need to examine this missing bits, whether it's 0 or not, because you can extend number of zeros. For example, 1. For example, you may be here, right? And if you have 4, this is in the middle. So how much you can check into the right? How much you can check to the left? If you have 4, for example, maybe you are here, right? So you missed this one. So you are not sure if it is 1 or 0. If it is 0, you should not replace, because these are more than 2 zeros. In this case, you need to check one further step to examine if you have 2 zeros or more. Now, if it's less than 2 zeros, that's fine, perfectly perfect. So you can still check the 1 0 and 2 zeros. Now, that's why we are telling you always you need to design an algorithm that is hardware aware. Now, since we have in mind that we need at least an FPGA with 5 input lookup table, so we have to target very specific FPGA, right? If we target FPGA with 2 input lookup table or 3 input lookup table, that is fine. Still might work, but you need 2 to operate in the same pattern. 1 operating here and 1 operating here. And the 2 lookup table in 1 pattern. But using 5 input lookup table, you need only 1 lookup table operating in each 5 bits. That is a kind of optimization. And for that information, we target the Xilinx Vertex 7 FPGA. And this Xilinx FPGA, they provide, this is the logic block they have, and you can read about the specification of the lookup table they have. So you can configure it as 6 input or 5 input lookup table. And if you configure it as 5 input lookup table, you got 2 lookup table within the logic block. So you are going to save a lot of resources by targeting this kind of FPGA. And this is what we did in the paper. So we target the Vertex 7 and we implemented, and we got this much of throughput, this much of speed up, and this is the setup, the complete setup, how we operate this as a read mapper. So we got the data, we do the indexing at the CPU side, we send the pairs to the FPGA, we do the filtering, and only the things that pass the FPGA go to the expensive algorithm. This is when we implement it. It's very cheap computation. That's why it does not occupy much from the FPGA chip. It's very small footprint. This is only gatekeeper, and that is for the PCI Express communication between the FPGA and the CPU. These are the results. The code is already public, available online. You can check. This is another implementation using SIMD acceleration. This is the paper for gatekeeper. Any question for gatekeeper? Okay, let's move. Now, can we do better? Can we solve the same problem, but in different algorithm? That is the different paper called Shuji. So what we did in this paper, let's go directly to the example, because I like much more. We have the first step is to build the neighborhood map. This is what we call. So in this way, we build similar to dynamic programming table, but without any dependencies. So we said compare the G with the G, compare G with G, G with T, G with G. Whenever it's hit, you have 0. Whenever it's missed, you have 1. This G is not similar to T, have 1, and so on. So you build the entire thing using this algorithm. What we do, we said have a search window of size 4 columns. So this is your search window, size of 4 columns, and then search for in diagonal way how many zeros you have. So check this diagonal, this one, this one, this one. Whenever you got zeros, highlight them in yellow. So count how many zeros you got per diagonal. So here you got 1, 1, 1, 1. Here you got 4. You can see them highlighted, 1, 2, 1. Now, since you got here the largest number of zeros, copy them as they are to the final result, as they are, even if they are 0, 1, 0, 0. If they are the largest number of 0, copy the entire content of the diagonal to the results. Move to the next search window. Again, 4 columns, size, and count the number of zeros. The one with the largest number of zeros has 3, so copy the entire content to the final results. Now, since the search windows are overlapping to each other, so every time we are going to overlap with the final results here, right? So what we do? So now the final results of this will be copied here and overlap with the previous results. Which one to keep, which one to consider? Are we going to copy always the recent results and overlap or overwrite the previous results? No. Our goal here is to maximize the number of zeros in the final bit vector in this one. We need always to maximize the number of zeros. So if we have 2 results overlapping with each other, which one going to give us maximum number of zeros in the final results. If this is more beneficial for us, let's consider it. If the other one more beneficial, let's consider it. Why is that? Because our goal is to keep all correct sequences. And correct sequences mean less number of ones in this final result. Yes, that's going to underestimate the final results a bit, but it's still providing great results. When we do the final comparison with Gatekeeper, this is providing great results plus great speed up. Why is that? Because we are dividing the problem into a problem of only 4 bits or 4 columns, right? So this problem is really independent from the previous search window, independent from any of the search windows in the table, right? All of them independent of each other. They only depend on each other at the very final stage, only at this stage, when we want to write the results. So we want to know which one maximizing the results. That's why we want to wait for all the threads or all jobs to finish and give us the final result. Then we decide which one to keep, which one to ignore. We keep doing that until we reach the end, and then we got this factor, we count the number of ones, and we compare to the threshold, and we see if the 2 sequences are similar or not. Very straightforward. Do you have any question? Okay, hopefully that is a good sign. Why we call it Chuji? This is the Japanese door where you slide it to open, slide to close. That is the word Chuji, so which is the door, because we slide the search window over the table, right? So now why we choose 4 columns? So we evaluate different number of columns. If 1, then we got very high false positive. We are going to accept all the wrong guys or the bad guys. As long as we increase the number of columns, then we got less number of falsely accepted pairs. And if we exceed the 4, then we start rejecting correct ones. So we stop at 4, and we said 4 is the right number for us. The nice thing, you don't need to build a counter. You don't need to build a counter for each diagonal to count how many zeros you have. Why is that? Because there are 4 possibilities, right? Or 4 bits, which is 2 to the power 4. That's the number of possibilities you can get. And that is easy to do using a lookup table. So you can look up these results, and any of them you can save how many zeros. If you got this pattern, this is 0. If you got this pattern, then it's 1, 0. This is 1, 0, 2 zeros, 1, 0, 2 zeros, and so on. So you can build a nice lookup table out of all these results without using counter. What's wrong with the counter? If you want to count 4 bits, then you need 4 flip-flops, right? Or D flip-flops, right? This is how you implement the counter. Then you need to wait 4 clock cycles to get the results, right? This is how we implement counter. So if you are waiting 4 clock cycles to get the results, that's a huge workload, because we are going to do this for the entire sequence, and the sequence can be 1 million long, right? So using the lookup table is going to give you the results just like that, right? There's no clock cycle. You don't need to wait for clock cycle. Only just to synchronize the results, yes, you need a D flip-flop at the end as part of the FPGA. That's why it's very efficient to implement this algorithm in FPGA. Now, maybe the last filter, and then we can move to another topic. Hopefully we have time. What we do here. This is similar to the dynamic programming table, which we call a dot plot or dot matrix. You can check this paper, who proposed the dot plot representation. Here, they have a region in the reference genome, and here they read itself, or it could be another region in the reference genome. In some application, you could compare genome to genome or read to genome. What we do, whenever you get a match, just base match, like this base is similar to this, then you record the dot over there. It's exactly similar to Shuji, for example, where you compare each character with other character, and you get this representation. Now, what we observe, if you consider very long sequences like this, maybe this is 2 million or so. If you consider very long sequence here and very long sequence over there, the result of the alignment is always scattered around the diagonal. This is the result of the alignment. And all of those just noise, because it happened that this character matching that character, because you always have 4 possibilities, either A, C, G, T. So the chance that any character that can match any other character is really high. So since we observed that the results always scattered around the diagonal, we said, let's solve this using very similar problem or very similar algorithm of finding shortest path. Think about this as a maze, and your goal is to find shortest path from this point to that point. If you find a path or such path, which is shortest, it means the 2 sequences are similar to each other. If you don't find such path, then it means the 2 sequences are highly dissimilar from each other, such that this path might go here, then there, then there, then there, something like that. Because any variation happening here will cut this line, and then you traverse to something else. If you see, for example, this is really opposite direction to this. It means there's inversion happening over there. There was an inversion in this region. That's why you got such plot. Okay, so if there are a lot of variation, then this line won't be around the diagonal. Will be traversed through different diagonals. So that is sneaky snake. We said we have the same problem in VLSI chip. So this is the VLSI chip. If you dissect through it, you will see this from inside. You have a lot of modules on the system, and those are pins for input and output. And your goal, you want to move from here to here. So think about yourself as a snake. You want to go from this point to the destination through these modules. Now, if you cross through too many modules, you want to traverse here, then stop, then go to somewhere else, right? Because there is an obstacle in front of you. And this is how we built this new algorithm. So we built this matrix first using this equation. You can read the paper to know how we come up with this equation. But assume that this is what we got, right? This is our edit distance threshold, 3. 3 means we are allowing not only for 3 obstacles or 3 errors, but we allow for at most 3 consecutive errors, at most. So you could get 3 insertion next to each other, but then at the other side, you got another deletions, 3 deletions, right? And so on. So 3 doesn't mean here the number of variation, but this is a threshold for the number of variation next to each other. So we build here 2e plus 1, if you observe that, based on the 3 threshold over there, and we convert each one into obstacle. Each one into obstacle. The 0, no obstacle. So you can see here in this location and this, it's 0, right? So we keep it as white box. So this white box, no obstacle. The 1 mean obstacle, so we cannot traverse through this path. And now our goal becomes going from here all the way to there. So this is the entrance and this is the exit. Now the snake, what we'll do, try to discover each of these paths. So here I have obstacles, so I cannot pass through. So let's ignore this path, go to the next. An obstacle, we cannot go through. Ignore, go next, next. Here I can go through. Only at this path, I can move 4 steps further, and here I can move 1, 1. So I just count how many steps I can move further or forward, and I find there's 4 steps over there. Now, I find the obstacles over there, so I have to iterate again, repeat the same algorithm. We count how many steps we can move forward based on the white boxes, and we say this one has the largest number of boxes. You can see that from Gatekeeper, Shuji, Sneaky Snake, that we always prefer to have the largest number of 0 in order to not overestimate the number of ones. We always want to underestimate the number of ones so that we'll be in safe side and we never filter out similar sequences. Now, at this stage, every time I find an obstacle, I reduce the threshold by 1. So it was 3 in the beginning. Here I reduce it by 1, becomes 2. At this step, I reduce by 1, becomes 1. Now I am allowed to avoid only 1 obstacle. So if I couldn't make it here, the snake will die, and then these 2 sequences are not similar to each other. So here I find the path, path, path. Which one to pick? Doesn't matter. Pick any path as long as they have the same number of zeros. So there are no ties here to break, basically. Why is that? Because we don't want to prefer one path over the other. We don't care. All of them will be penalized for one edit since we have an obstacle. So we choose any one of them and we reduce this by 1. Now the 1 becomes 0, but we already arrived to the destination within the allowed number of, or within the threshold. I shouldn't say within the allowed number of errors, because as I said, this is not number of errors, this is just the number of consecutive errors. It's different meaning. In reality, I don't build that. In reality, what I build is this. Why? Because whenever I face an obstacle here, I don't need to continue calculating this. Whenever I face an obstacle, don't calculate anything. Just calculate whenever you see no obstacles. And that's why this is very cheap. This is the fastest so far. We proposed it around 2019 or 18. And still, until now, it's the fastest realignment filter ever on CPU, FPGA, and GPU. Because it's really small architecture, small data structure. We don't build the entire matrix as in Gatekeeper or in Shuji. This is only the small part that you need to build. And that's why it's really fast even in C programming. You don't need to do AVX acceleration and such. The resource utilization of the FPGA and GPU were really small too. So these are the benefits you can get out of SneakSnake on real setup. So compared to Paracel, EDLib, which were the state-of-the-art, KSW2, which is still state-of-the-art sequence alignment. So we accelerate sequence alignment by this much, which is really impressive. Now, coming back to the data movement problem, we already explained that we have a serious problem with the data movement. We said, let's use processing in memory to solve these issues. So you already been introduced, yes? That's really great question. So wait until the end. There is a work called Gen Cache. I'm going to explain about that. This is the exact idea they follow. This is not from our group. This is from different university. All right, so Juan was explaining a bit about in memory or processing in memory. He's going to introduce you more into real processing in memory device from UpMem company or French startup in the next few lectures, but there are already existing lectures, you can watch them. And then let's talk about this paper. We observed that still, even if SneakSnake is fast, but the data we were moving from the CPU to the FPGA were bottlenecked by data movement. So we were using PCI Express 3 at that time, but was not fast enough. And in these days, we have 4 and 5 versions of PCI Express, which were much higher bandwidth than what we used in the past. So we said, let's perform read mapping near where data resides. Rather than moving the data from the memory to the FPGA, let's do the processing inside the memory or very close to the memory, using very short wires from the memory itself to the FPGA logic. And that was possible only because if we have real devices from Xilinx, where they have HBM memory, high bandwidth memory, integrated on the same package with the FPGA logic that we have on the board. So when you have them integrated in the same package, it means shorter wires to transfer the data, then less energy need to be consumed and so on. And you have also high throughput, since all of them in the same package. You don't have PCI Express or whatever links to move the data from one point to another. We also try another FPGA, that is the traditional FPGA, where you have DDR4 RAM, where you can move the data, but the DRAM is separated from the FPGA itself. They are not on the same package. The benefits we got were really impressive in terms of execution time and energy efficiencies. We were having some lag in the performance just because of the data communication. We were using OCAP interface and CAPI2 and so on. So Gagang is here, who was leading this work, and maybe he will going to explain about it in the next few weeks. Let's see. But we improved the performance of SneakSnake just because of the technology improved, just because we are able these days to get high bandwidth memory coupled with the FPGA in the same package. That was the work. in the past, we target 3D stacked memories. So 3D stacked memories, basically this shape. So we have a logic layer that you can configure, you can program, same as Fpga, for example. And you have a different layer of DRAM. So all those are memories. They are always stacked on top of each other on the same package. So again, here's different from the HPM Fpga setup that we were using. But this is no longer exist, at least for HMC cube, for example. But you can simulate the setup. You can assume that there is a logic layer. You can assume that there were this many of DRAM layers on top of it. You can assume the communication between them is this much of high throughput. But we don't have a real device that can emulate this structure until now. Now, if you want to exploit such architecture, you cannot fit Sneak, for example, as is, or Gatekeeper as is. Why is that? Because, sorry? Okay. Yeah, so we need to change the algorithm. The way we deal with the data, we need to really change it so that we can leverage and exploit the entire architecture. Let's see how we leverage it. But one more point, that you can access these layers vertically. So you can access this bank, fetch the data, and move it direct to the logic layer. So the more you can fetch vertically, the better you can process at the logic layer, and you can parallelize the operation. So what Jeremy did, who's the first author of this paper, he said, okay, we have a reference genome. Now we don't want to get k-mers, but what we want to do, representing each region in the reference genome using a bit vector. So use a bit vector like 500 long, for example, or 512, because we like multiple of 2. And then we want to represent the entire region with that bit vector. And whenever we get this bit vector or similar to this bit vector somewhere else, then we said they are matching to each other. So there is no k-mers here. We got bit vector, 500 bits and 500 bits, and we always compare the 500 bits together. So what we store in the DRAM layers is the 500 bits representation of the reference genome. There is no hash table, there is no indexing here, which is basically kind of indexing, right? Because you consider a chunk of the genome and you convert into bit vector. Whenever we got the same bit vector or with differences, then it means they are similar to each other. How we build a bit vector? We said, let's permute all possible values of 5 characters. So AAAA, AAAC, all the way to 5 T's. So let's calculate all the permutation of any word of length 5, right? Because we only have ACGT. Whenever we got this appearing in this region, we record 1. Whenever we got this appearing, it doesn't matter where it appears, first or second or third, doesn't matter. We record it just as 1 in the same location. So this is already sorted bit vector. We cannot record this value, for example, here. It should be in the same location. Now, we got this as the final result, and this is what we store in the 3D stacked memory. Now, this is a one-time process because we do it for the same reference genome. As long as you are using the same reference genome over and over, then you don't need to rebuild again. But what you need to rebuild for the read, because the read came in real time. You cannot pre-process it. Now, if we got another read, this is the reference genome, for example, and this is the read sequence. We do the same, and what we compare in the logic layer is this bit vector to that bit vector. How many ones we have there? How many ones we have there? Then if the number of ones are similar here, not equal, because we need to allow for some tolerance for edits, right? Because if we are missing 1 here, it's still similar as long as it's within the same threshold. But now, is it only about the number of ones or also the location of the ones? We need to consider the location because the location in the bit vector has a meaning here, right? It means that chunk already exists. And if that chunk also exists, so the corresponding one should be the same. So it's kind of similar to the hash table because we have k-mers at the end. But the nice thing We could easily fit bit vector in the 3D stacked memory, but we cannot easily fit hash table in the 3D stacked memory. That's why we always need to build an algorithm that is aware about the hardware. So once you know what is the hardware requirement, then you can design your own algorithm. So let's skip these details. This is how we integrated with the full system and what are number of resources that we need from the logic layer to implement it. I'm going to direct you to the video to get more details about this work. This is from Jeremy, who's the first author of this paper. And this is the paper itself. We have the GitHub code online as all our works, so you can easily go and leverage it. Now, coming to your question. This is Gen Cache, where the key idea, they have two key ideas. The first one, there are huge data movement between the CPU and storage. Let's do the calculation inside the cache. We don't need the CPU to do filtering. Let's do all the kind of filtering inside the cache itself. The second thing, that let's not use the filters separately from each other or one filter. Let's use a bunch of filters because each of them are good in detecting type of dissimilar sequences. There is no one best realignment filter at that time. So this is before Sneaky Snake. So now maybe with the Sneaky Snake is only enough to use it all the time. We don't know that. We need to evaluate. What they do, they said it's beneficial to have, for example, Hamming distance when we have error 0. So when the user said, I want no error at all, I want exact match, so let's enable Hamming distance only. But when the user wants to allow for one error, let's use this algorithm. When the user wants 2 to 5 errors, let's use these algorithms all together. When I want to allow 6 or more errors, let's use these algorithms together, and so on. And that is a nice thing when you know the algorithm and the capabilities of each of these algorithms. If you know that your algorithm is capable to do that, then it's good to target only these specific operations. But still open question. We don't know what is the best combination of algorithms to be used. So it will be a nice project if someone can come up with those evaluations. All right, so now I think you are going to be introduced to this work, GenAsm, CGRAM, GenStore, GenPip in the next few weeks, but I'm going to flash them just quickly for you. So GenAsm is one of the recent work that we did 2 years back. So our goal was to accelerate sequence alignment, not pre-alignment filtering, sequence alignment, the dynamic programming itself. And what we do, we said there was a very old algorithm called BiTAP. It was proposed in '92. And we said this algorithm has some drawbacks. So it cannot do these things, cannot be parallelized, cannot do traceback. What traceback does mean? It means when you have the dynamic programming table, you want to fetch the final result to say how many errors, what are the type of errors, and so on. That is a traceback step. So you need to traverse through the dynamic programming table after you solve it. After you complete everything, you start traverse in the opposite direction. So you start from the top corner all the way to the bottom. Once you fill everything, you start now in the opposite direction, from the bottom to the top, to see which path you were following when you were filling the entire matrix. And that is going to give you more information about the alignment. It was not supporting long reads. So in that work, we solved all these drawbacks of the problem, and then we target 3D stacked memories for the acceleration. So I'm going to flash the results. We compare it with the state-of-the-art work, Minimap2, BWA-MEM, Darwin, and GenX. We also compare with pre-alignment filters, since GenAsm, let's say, accurate sequence alignment, right? And Shuji is a pre-alignment filter. So we want to say, what is the benefit if we make sequence alignment 100x faster? Do we still need pre-alignment filters or not, right? So the speed up were kind of good, 3.7x, but that was only for short reads. We don't know for long reads whether sequence alignment make it more efficient, more faster, will still be better than pre-alignment filter or not. That is a full video explaining the entire work, GenAsm. So please check it out if you're interested. Now, GenStore is a more recent work we published on March this year, and that is processing inside the storage. So we said there are two types of data. That's why I encourage you in the beginning to know what you are handling. So you have to know the properties of the data that you are targeting. So we know there are two properties. The first one, we're targeting either short reads or long reads. The short reads are accurate all the time, 99.9% accurate. So they are exactly similar to what you have in regions in your reference genome. So we said, if we find these reads that are exactly matching to your reference genome, there is no point of doing indexing and sequence alignment and all this stuff. Why? Because they are exactly appearing in your reference genome, and there is no variation at all in most of them. Of course, you will get variation because we are always comparing to a reference genome that does not belong to us. It belongs to someone else. And there are always differences between the sample and this reference genome. Now, the second idea is the long reads. Now, long reads is totally different story. We cannot say we always expect to have exactly matching reads, right? Because in long reads, by nature, there are a lot of sequencing errors. That is one. Second, there are variations since they are long. They are spanning longer regions. So we always expect to have variation within these reads, not as in short reads. So we said, let's design two different algorithms and two different architecture depending on what data we are targeting. The first one, if we are targeting short reads, then we will design exactly matching filter. So we built a huge hash table inside the SSD system. So we have pages, and each page we represent a chunk of the hash table. And we make the hash table over there sorted since we sequentially can access the page. So we sort the hash table, we save it there, and what we store inside the hash table is the entire read, not seeds. So it's kind of similar to the indexing, but now we hash the entire read, not only seeds. We hash the read, we store it, and then we check that hash table quickly in the SSD system. If we find it, it doesn't mean it's noise or we need to delete it. That is a very good output. So we need to keep it in the SSD. And once the read mapping done in the processor, we can merge these results with the output of the processor or the read mapping. Because these mean very good for us. They're high quality mapping. So each of the read exactly match to the reference genome. Now, in the second case, when you have very long sequences, again, we cannot use the same methodology for short reads. What we do, we said long reads, if they match to a region, then there should be, or they should share a very large number of seeds with that region. So we build a hash table, but this hash table has seeds, not read. So we don't hash the read, but we hash seeds now. And we count how many seeds we got from the hash table inside the SSD. If we got a large number of seeds, then probably this read is good. And now we don't do same as short reads. We need to send it to the processor to do dynamic programming here. So we do the filtering here for the reads that share a large number of seeds, and we send them to the processor to do dynamic programming. And any read that does not share a good number of seeds with the reference genome, we delete it inside the storage. In this way, we save a huge amount of time and energy by not moving the bad reads into the processor or to the memory, then to the processor and so on. So we filter the good guys from short reads here, and we filter the bad guys from long reads also inside the storage. These are the results. If you're interested in numbers, you can see the saving in terms of speed up and the energy saving. Any question? Okay. Yes. Yeah, I'm not aware of any. I don't think anyone has done it before. There is one work from Barcelona Supercomputing Center, but that is more generic. I don't think they solved the problem for read mapping. Yeah, they enable some computation near the SSD for genomics, but simple operation, such as exactly matching reads, for example, and such. But I know there is a company try to enable processing inside the SSD. So they integrate FPGA logic or chip within the SSD so that you can program these things. For us, we cannot implement it in real life. The GenStore work, we need some overhead or some extra hardware to be integrated with the SSD to implement it in real life. So I think those kind of companies who provide configurable computing next to the SSD might implement GenStore and have it as a real product. Okay, hopefully this is the last work called GenPep. This is very recent from our group. We just published it in Micro this year. What we do here, very different than the other work. We said there is a huge problem starting from the sequencing machine itself. It's not just when we map the reads. So there is sending the raw data from the sequencing machine, which is electrical signal in this case, because here we are targeting nanopore devices. So we are getting the electrical signal, and then we want to base call it. We want to check the quality of the base called read, and then we want to map those reads. And now, if the read is bad after read mapping, then we already wasted the time for base calling, we wasted the time for a quality check, and we waste the time for read mapping. So the bad read already passed from the sequencing through 3 steps, and at the end we deleted, right? So we wasted those cycles. So we said, why not examining the quality of the read much earlier than waiting for read mapping? So we have base calling, then quality check, then read mapping. And the nice thing in base calling, it's normally neural network that check the translation between electrical signal and the ACGT data. And what they do, they process the read in chunks of 300 long or 400 long. It's still configurable, you can configure that, but at the end, this is already existing. The base caller that tightly coupled with nanopore sequencing machine process the read in chunks. So we exploited that observation and we said, let's get the first chunk, it's already base called, and let's try to check the quality of only that chunk. And once it passed the quality, let's map only that chunk and see if it is mappable or not. If it is mappable, then that is great. Then we can continue base calling, and then we quality check and read map the entire read over here. But if at this stage or at this stage or at this stage, we figure out that the rest of the read is not mappable, then it may be a wise option to stop performing these 3 operations and just ignore the rest of computation. And this is what exactly we did. So at base calling, we wait until we base call at least 1 chunk, then we check the quality, we check read mapping, and then we decide, continue, no, something like that. And all of those are pipelined together. So we don't need to halt the operation at any of these steps. So you still, for example, while you are checking the quality, you are base calling the next chunk. And while you are read mapping, you are still continue base calling. And that is the nice of parallelizing or pipelining the entire system. So I'm trying to cover here many ideas so that you can not be scared anymore from this field of research. This genomics, still, if you don't think about the data itself or what is biology and blood sample and disease and all this, it's just designing algorithm as if it is any general computer science problem, right? So I'm going to flash now more papers. This is gatekeep that we explained. There is magnet, another filter, Shuji, Gram filter, and this is the HPM acceleration, GNASM, CGRAM, and this is another work in memory processing for microbiome profiling. And this is another work using real processing in memory. So this is the first real device that we target for the in-memory processing. So this is using UpMem system for accelerating wavefront, which is the state-of-the-art sequence alignment algorithm. That's in summary what we have done so far, a lot of acceleration work for different parts of the compute system. Now we have 10 minutes, and I think I can finish within the 10 minutes. Let's see. So as a conclusion, realignment can be substantially accelerated. Hopefully I convince you with that as well. Computationally inexpensive and accurate pre-alignment filtering algorithm designed for specialized hardware. We really don't need to perform sequence alignment all the time. And all the three directions that we explain are used in today ReadMapper. Although they are proposed 5 years back or 2 years back, they are currently used in ReadMapping, including indexing, seed filtering, pre-alignment filtering, and sequence alignment acceleration. The pre-alignment filtering, the nice thing about it is does not sacrifice any of the aligner capabilities because we don't touch it. We keep sequence alignment as is, and we have a process before it. Just decide when to use and when to not use sequence alignment. But we never compromise any of the capabilities of the sequence aligner. Okay, now let's just give you more imagination about things that you can do. It's not only read mapping. There are a lot of steps in the pipeline. That's why we call the lecture title Intelligent Genomic Analyses. It's not single analysis. There are a lot of analyses depending on the application you are targeting. Now, to refresh your mind about this problem, you have a reference and you have a pieces, you want to build them up to compare them to the reference picture. What if a country that is rich and they build many references? Because as I said, if you compare European guy with African guy, it really is not fair because they have totally different DNA. So it makes more sense to customize the reference picture. Now, what if you get different reference genome? Because even the reference genome from NCPI, every once in a while, they keep updating it. Now we got, I think, 38 version of it. So there are different 38 versions. Each has some substantial differences with the previous version and so on. So what if we got a new reference genome? Do we really need to repeat the entire read mapping process, assuming you already mapped those reads based on this reference? What if you get this guy? Do you need to repeat the things again? There are a lot of work showing that using one single reference genome for the entire world is not a fair game, especially for African. They show that there is 10% more DNA compared to the others. There's another paper showing that it's time to change the reference genome. That's why here we start talking about pan-genomics. It's no longer genomics, it's pan-genomics. What's pan-genomics means? It means a collection of people. Now you are not studying single reference genome, you are studying 10 individuals at the same time. So we did this recent work called airlift, and what we tried to do here to solve this problem. Now you have two reference genome, you already mapped to one of them, how you can leverage this existing mapping results to update your results for the new reference genome without repeating read mapping at all. What we do, we said, let's get the old reference genome and let's get the new reference genome, assuming both of them for the same species. So both of them for human, for example, or both of them for viral or same viral. So now we said, what are the similar regions between the two reference genomes? If this is similar to this and this is similar to that, then the read were mapped to this region, they probably will map to that region as well, right? So no need to repeat the read mapping process again here. Any read mapped previously to this region, let's just move it to the new reference genome. That's it. Assuming this region is exactly the same as this region. Now, there are some regions will be updated, right? That's why we got a new version of the reference genome. In these regions, we can do remapping only or update the results. If we know, for example, there is only single change here, so we only update for that single change. If this is a new region, then we need to repeat mapping most of the reads to that new region only instead of mapping to the entire reference genome. So there are a lot of nice things you can do even apart from read mapping. And that was airlift paper. Now, I want to talk about another, I have another lecture after this, but I want to talk about different topic, how we can enable the reuse of all those hardware acceleration jobs or the adoption of hardware accelerators in genome analysis. So one of the paper we published in December 2016, we got one review from the reviewers, because normally before you publish a paper, you send it to a journal or a conference, and then they assign 6 reviewers to your paper, and they give you comments on your work. If these comments were positive, then they accept to publish your work, and then you can go to a conference and present it. And this is what we got from one of the reviewers, which we don't know, because most of those reviewers are, it's a blind process, basically. So we don't know them, and sometimes they don't know us, or they don't know us, or they know us and we don't know them. So we don't know who wrote this review, but we'd like to thank him because we're very fruitful discussion over there. What he said or she said, there has been little to no adoption of previous specialized hardware solution related to improving the speed of alignment. And while there has been considerable work in this area, it does not seem that these hardware-based solutions have gained any type of real traction. So you are designing hardware accelerators and nobody used them. What is the point? This kind of what he's saying. And this is what we reply back. It always takes time to adopt new or different hardware technology, since it requires investment into the hardware infrastructure. So in that time, nobody was using Fpga in real life for genome analysis, right? All of them were using computer. That is in 2016. Now, I already attached our very long response in the slide, so you can check the slide later on. But in that time, we were dreamers. So we were dreamers, and the things came true after some time. So after we start releasing Fpga accelerator, then this Illumina itself, who creating the sequencing machine, they start using Dragon FPGAs, which they are customized for read mapping, filtering, and all this job inside their sequencing machine. So this is in 2018, they start having those accelerators. NVIDIA as well in 2020, they start having the Parabricks to accelerate inside their sequencing machine using GPU. However, all this works, since they are using FPGA and GPUs, you still need to move the data from the storage to the main memory to the accelerators, right? So hopefully in the next few years, we will start seeing more works on in-memory processing inside the accelerator, inside the sequencing machine. Those are more challenges on how to adopt the hardware accelerators. Coming back to our story, did we achieve our goal? I would believe not yet, but we are very close to it. Since all this work that we did, we tried to make the things more efficient, but I believe the best place where to do all these processes inside the sequencing machine itself. In this case, only we can have one portable device for doing the sequencing and for doing the analysis itself. We still have this open question, where and how to enable this intelligent genomic analyses. We start seeing specialized chip, a huge chip actually, to do machine learning, which is much larger than the capable GPU. Why not having the same for genomic analyses? In Tesla, for example, they are willing to build this huge board just to do the processing and self-driving, and even they have redundant chip, the exact same chip replicated just for safety. In case of failure, the second one will work. This is the sequencing machine from Illumina, where they have the FPGA accelerator inside it. And the very great news that we received this year, that NVIDIA, starting with the H100 GPU, they have one module just to do dynamic programming. Just one module to do sequence alignment. You can see the importance of this problem. That is something really great. So you're solving sequence alignment using a customized hardware within the capable GPU. And that's going to provide you, as they claim, 7x of improvement using this capable GPU. That is something nice. If all these companies start producing specialized architecture for genomics, then we may enable more things. We recently received this grant by UPM to discover the possibility of using this real hardware. This is from UPM. This is typical DRAM. So this is same DRAM you have in your computer, but the difference that you have DPU inside it. So the DPU, where you can do processing inside the DRAM itself. So you have the memory banks, you move the data within the bank to the DPU to do processing inside the DRAM. And this grant is about reusing this real hardware to do acceleration within the DRAM module. So we are going to do or evaluate different algorithms, different application for genomics, and see whether they fit in this real hardware or not, and how much we can accelerate the things. Okay, that is the last question here. What if the sequencing machine, instead of giving us chunks of your genome, they now give us the full genome, the exact full genome as is? What will happen to all the acceleration work, all the effort spent in the last 20 years, for example? So there are a lot of fields different from read mapping, for example, metagenomics, pangenomics, where even if you have the entire genome, you still have the question, which genome is this belong to? So the sequencing machine will read the genomic data, but cannot tell you what species is this. So you have the full genome, and you still need to compare it with all existing genomes that you know, such that you said this genome belongs to this virus, or this genome belongs to this bacteria. So you still need a lot of techniques to compare genome to genome, for example. Same in pangenomics. Even if you know, like the genome of 100 individual, how you can compare your genome to those 100 individual genome, which is basically represented as a graph sometimes. So even if we got such sequencing machines, still all the work that we propose or other work from other groups can be reused, basically. So hopefully we introduce you to system design for bioinformatics, which is a really critical problem. It has large implication on everyone, basically. And the lecture was about accelerating key step in bioinformatics, which is read mapping, but we also talk about base calling and variant calling, quality control, and so on. There are many bottlenecks, some of them general to the problem or genomics, and some of them really specific to each step you're handling. And we cover various recent ideas. Hopefully, most of the work that I covered published in the last 2 years or so. And this journey, we started as Safari Research Group in September 2006 with my PhD advisor and Onur Mutlu, where they were working in this area in the past. And they continue working on it, hopefully for the next few years. The takeaway, population scale analysis is not easy task. It's never been easy as well. You need to consider many things in designing a new system. As we said, you need to know what hardware you are targeting. And you know, when you implement it in hardware, your hardware needs to be aware about your algorithm. It was fun while we developed such thing because we help in genomics, which is affecting our health. So it's really novel and nice to work on this area. We enable, hopefully, a great future for everyone. It's a very hot topic for grad studies. We have a large number of students in Safari Research Group just focusing on this domain. Regardless if you are targeting FPGA, GPUs, any kind of platform for acceleration, this is a very good topic for you. Because at the end, you need application to show the benefit of your architecture. Most of the speedups come from parallelism enabled by architecture and algorithms. Would like to acknowledge all the authors of the work I presented today, including Onur Mutlu, Jana Lakan, who was my supervisor during PhD, and our collaborator, Sergey Mango. There are a lot of readings in the slides, so hopefully you can check them, especially the first paper that we show it to you. There are a lot of videos you can go over once you need more information. And we are 1 minute late. Okay, thank you so much for today. And if you have any question, feel free to send me directly, or we can discuss it in the next lecture. Thank you. Hello.